Hello. All right, the last couple of assessment statements for topic 2.6. So one is just draw and label a diagram. So I believe I've given you plenty of practice on that. So please practice drawing it. And again, I do want to stress that it's very important that you're reading it carefully. So they might ask you to draw a single strand. Make sure you're counting how many nucleotides they're asking you to draw. So single strand would just look like this, right? One on top of the other. Or, so that would be two nucleotides attached in a single strand. Or they might ask you to draw a double strand. And if it's a double strand, then you would draw one on this side. And remember, your pentagon has to be upside down when you draw it. Okay, so go ahead and practice those. All right, still haven't figured quite how to get to the next slides seamlessly. So 2.66, in this one, they want us to compare DNA and RNA. Compare again means your similarities and your differences, and the best way to do that is in a table format. But if we're looking at our DNA and RNA, if we just start with the sugar, so here is our 2-pentose sugars, right away we can see that there's a difference right here, and that is actually the only difference. So because the hydroxyl group, hydroxyl is our OH, because the hydroxyl group is missing and replaced with a hydrogen, it's called deoxyribose. Whereas on this ribose, which is the ribose that we studied in carbohydrates, has that carboxyl group there. So that's one difference. However, a similarity would be that they both contain phosphate, right? There's phosphates coming off carbon number 5 on both of those. Not the greatest circle, but so phosphates are coming off both of those. They also share nitrogenous bases. They both have, it's hard for me to see, cytosine. So that's RNA is on the right hand, DNA is on the left hand. They both have guanine, and they both have adenine. So those three bases, they have the same. However, the difference with RNA, instead of having thymine, it's replaced with this other type of nitrogenous base called uracil. So that would be a difference. Also, the structure itself, right? RNA is made up of one backbone, one strand. So it's not a double strand, not a helix. So that would be another difference. So I've organized that in table format. If you look at the shapes or the structure, so double helix or single strand are bases that are the same and different. I recommend doing bases that are the same. So ACG, ACG, and then the difference being uracil and thymine. Our sugars are different, deoxyribose and ribose, and also the lengths are different. Not always, but they can be in more complex organisms like ourselves. They can be quite longer than the DNA can. The last little piece, and we're going to practice this a little bit in an activity later. The last little piece, and there's a lot on this. There's a lot on this text. So, or on this slide, a lot of text on this slide. So they want you to understand the importance of modeling in science. So just the main point here is that Crick and Watson are famously known for discovering that DNA is this double helix. So it's like this. And not only that, they're joined together by bases. And they, through modeling, were able to figure out that adenine, will always form complementary base pairs matching base pairs with thymine, and the same with cytosine and guanine. I'm doing 3 lines here for my hydrogen bonds. And they did this just by knowing the molecular structures of these molecules, and by trying to squeeze them together almost like a puzzle piece, they were able to figure out like how they would actually form. And this is an actual photo of their model. So they use these wires, and you can see the bases. Here's our pentose, right? But you can see the bases in the middle here, and they only fit together in this very specific way. But I have a nice activity that's going to summarize all of this together, and hopefully you can visualize that better. So that is it for 2.6.